thing on. You guys hear me? Okay, so we are just about ready. It's going to be navigating grid cells and place cells. Should be an interesting talk. We're just waiting for someone to join the meeting. We're all working from home right now because of the COVID-19 thing. All right, here we go. We're going to get started, I think, real soon. You guys ready?
3, 2, 1, let's go.
Okay, go.
All right, so there's no on-air sign, but I assume we're on air, right, Matt? All right. So Subutai pointed out this special issue of Hippocampus Journal, and I looked through it a bit. I read the introduction, some of the articles, but he pointed out this one in particular, which might be interesting to read, and I thought it was really interesting. And so I just want to talk about it. In some sense, it's a beautifully written paper. It's very easy to read. It's very understandable. And so I appreciated that very much from the authors. It's about rat navigation, but I think there was some real insights in it that just things I hadn't really put together before. So a few things that struck me as like, oh yeah, that makes sense. Oh yeah, I sort of knew that, but I didn't think of it right. That kind of stuff. So I'll try to highlight that. Although when we go through it, it's going to be a lot about rat navigation. But again, I'll try to point out the key insights that I got from it. And of course, if anyone else wants to jump in, go ahead. Essentially, what they're talking about here is how, of course, rat navigation is how a rat gets from someplace to someplace else. It's just typically where they are and a goal, direction they want to get to. And they make a very clear distinction between two different strategies for doing that. And we've always talked a lot about how grid cells can provide a sort of vector-based method or a metric system to say, okay, if I have a metric space defined by grid cells, I can say, take my current location and calculate the direction and distance to the goal. And that's just like a straight cross shot to that goal. We've talked in the past about mechanisms that are proposed for that, but they're kind of wishy-washy. But that's one idea. And the other way of navigating, which I hadn't really understood before, is using place cells. And this is, they call it a topological strategy. So in this highlighted text in the abstract, we have place cells have been associated with topological strategy for navigation. Grid cells have been suggested to support vector navigation. And the idea with using place cells for navigation is you just retrace your steps. So if I've gotten from A to B and I want to go back to A again, I can retrace my steps using the places I visited. That would be the topological strategy. And the vector strategy, say, no, I just go straight from B back to A. The figures, I've highlighted some text in here, but I'm just going to write to the figures because I think the figures are very nice. They support it. This is figure 1. It shows basically the two basic strategies. They're showing it here, a little picture of a house and nest or something like this. I guess the rat is visiting the home to get some food and going back to its nest. I don't know. And they show the little path of the rat traveling along this dotted line. And then the rat is supposed to be at this current location, the back of the building, and it wants to get back to its nest. The two strategies in B, it shows like, oh, we could just follow the link of places it's been to. And so the idea here is, and I didn't really understand this, but a lot of people believe that place cells are linked together sequentially in series. So it's like a sequence memory. And you just follow the sequences of the places you've been, but going backwards order. And of course, on the right here, we see the vector navigation, which the rat says, oh, I can calculate the straight path to get there. And what this paper is about how to combine the two of these strategies, that both of these strategies may be deployed for an animal trying to get someplace. I won't go through the rest of this figure. I think it's more useful just to go right on to figure 2, which shows the basic idea here. So what they are proposing is the following, is that an animal starts off with the vector strategy, which is grid cells, a metric space. And so, and it starts heading straight towards where it wants to go until it gets to an obstacle, and then it decides what to do. when it gets to an obstacle. They have proposed a simple way for the use of border cells. So here you're showing an animal that is let's see here. He started out here on the right on this A figure. He's now over here and he wants to get back. And so using the vector strategy, he goes straight towards the original spot and he runs into a wall, and it's a slanted wall in this case. And what they're proposing in their model, this is all, they build a model of all this. What they're doing is the animal starts with a vector strategy, and when it gets close to a border, the border deflects the vector in some sense. And so here they're showing here's some motor cells that are indicating the direction the animal is supposed to go. But when it bumps into the border, there's an inhibition of certain of those, the motor cells that are active. So the ones that are, you can see there's a motor cells that sort of mostly to the right, some up and some down. That's a distribution there. But when it gets here, this red inhibition deflects the motor cells to affect more to the left or up in this picture. And so as the animal gets closer to the border, it bends around and it will just go around the border and then continue on to the goal. And so that's part of their model. It does that very nicely. And then, but there's a problem if the animal runs into an obstacle, that strategy doesn't work. So in this case, the animals come around here and wants to go back home, and it bumps into this vertical wall, and there's no clear strategy. This deflection method wouldn't work here. And so what they're proposing happens is that when the animal gets to this point and it stops moving, it says, okay, I'm stuck. What do I do? There is the replay of play cells. Now, this is something we've heard about in the past, which I never had a model in my head what the replay is for. So just review what's going on here. When sometimes an animal stops moving, they see that the play cells very rapidly recall a sequence of recent movements in the exact path. And so what they're suggesting here is the animal, when it gets stuck, starts recalling how it got to its place over here on the blue dot. I hope everyone can see my cursor. Can everyone see my cursor moving? Does it work? Yeah.
Yes, we can.
So this happens very rapidly. You know, there's a very sequence occurs in this 120 millisecond burst of activity that it starts playing back the play cells in order. And as it's doing that, it's calculating the play cells play back, and they argue that the corresponding grid cells play back too. And so it's basically can recalculate a new, from where it is right now, stuck at this wall, it calculates a new vector direction based on the grid cells. So, and as soon as it sees, oh, I can get to this new target. The new target is over here now in this third diagram. He says the new target's down here. He says I can get to that. So it just basically says, okay, now this becomes my new temporary goal. And once it gets to its temporary goal, it goes back to doing vector navigation again. Says, oh, I can go straight to the target again. So there's this interplay between first starting a vector navigation, deflecting around obstacles that can be deflected around and still keep going. But if you get stuck, then you replay very rapidly. The animal stops moving. It replays sequences of how it got to where it was until it finds a new goal and then continues on from there. This was the first time I had ever heard of a functional role for the replay sequences that have been observed in play cells. And I wasn't aware that those replay sequences also invoked grid cells in the same path, in the same way. And so I thought that was very exciting. And, you know, because one of the reasons I've been thinking about this is when we think about a cortical column, in some general way, very simple way, you can think about the upper layer cells as being like play cells. They are what's sensed in the environment of the world of the animal. And we've talked about how to learn sequences of those inputs, but we've never talked about learning sequences of sensory motor inputs. And essentially what's happening here is that they're arguing that even in a sensory motor situation, when the animal originally moved from this red circle all the way around to here, it was remembering in one shot learning, it was remembering that sequence of events. And our sequence memory could do that. We haven't thought about doing that because in general, it's not a very useful thing to do. If you're trying to learn a model of an object, it's not useful. But if you're trying to navigate it, it is useful. So I thought that was pretty exciting idea. Hey, maybe we're doing this one-shot learning in the upper layers of the cortical column, and it could serve a similar purpose here. Okay, so that's it for that figure, unless there's questions about that.
I'm just thinking it might be helpful to actually play the video.
If you got it, I don't have it up on my screen.
Yeah, I got it. I don't know if I can briefly take over the screen.
Do I have to give it up?
Yeah, you should just be able to share it, I think.
You cannot start screen share while the other participant is sharing.
I can stop my share.
Right. Because it's kind of cool to see this stuff dynamically.
It's funny. It's funny. I had no interest in seeing the video because I think I understood it completely from this figure, but it might be interesting to see.
Right.
Jeff, how does it figure out what those, where to place those little triangle landmarks? At the bottom of the right figure, bottom right figure, those green triangles. How does it know what those intermediate goals should be?
It basically as it's playing back in the middle figure here, as it's playing back the place cells. So the red circle goes from here to here to here to here to here. They're just showing it in one step. It keeps going, and as they're going, they're constantly. Their model says that every single step along the way, they're calculating a vector, and there's a strategy they're calculating a vector from where the animal currently is to where the red circle is. And there's a little controller, which I didn't look at the details of, to decide when has the red circle gone far enough that they feel that they can stop doing this temporary search for a new goal, and then just switch to the vector navigation. So it seemed to me that even when the red circle was here, the deflection strategy would work at that point in time. The animal could say, oh, I can start moving in that direction. That's not the way they described it. They described it, they waited till the green triangle, till the red circle got all the way to this clear shot. And once they got a clear shot, then the animal says, okay, I can get there clearly. I don't have any obstacles in front of me. I'm going to go. I wasn't clear to me why it had to be that way. I thought that why couldn't the animal start heading towards this goal right now? Because it's in the deflection scenario that would work. But anyway, there's a mechanism which determines when to stop moving, when to say that's enough replay. Let's pick a new target, which is the green triangle, and now switch back to vector navigation. Did I answer that question?
Yeah, sort of. There's some sort of an algorithm for doing it. Maybe
they have an algorithm. Yeah, I didn't like.
If you know the angle of the border, you can probably figure out once the vector is lined up with the border, then maybe you have a clear shot.
Yeah, but I didn't see why they had to wait for a clear shot.
Because at the middle, at that point, it looks like the slanted one up top.
So I think that wasn't the key part of their model. I think the key part of the model was that there's these two methods of navigation, and here's how they alternate back and forth. And they had to make up some fudgy sort of I think they had a name for it. I forget what it was called, but they had a name for this algorithm. It's decided like, how long do I keep doing this playback until I decide to go back to vector navigation? And I'll talk about it in a moment. If they also said, if through this process of playback, there was never a place you could go, like you're totally stuck, then the animal just has to go walk its way backwards. They basically. And it updates its model too, which is interesting. We'll talk about that in a moment. Yeah. Yeah. Yes, thank you. Right. I knew that. I think what was interesting to me, because there's a lot of literature about this. I'm sure the author, which author who's on it? Okay, thank you for watching.
Hey, Andre, nice to see you again.
I hope I'm not misrepresenting your work. It makes me nervous now. Okay. Right. Obviously, the replay has been implicated a lot in memory consolidation and I think some other things. This is the first time that I was aware of it providing this sort of functional, really tangible functional role in a cognitive task, which I thought was exciting.
Yeah, I guess the key thing really is that they take the grid cells along during the replay, which allows for the continued vector navigation. So you don't have to choose between two strategies, but you can actually alternate between them. Obviously, much of that is based on that very brief Ólafsdóttir paper, which I still have a lot of questions about in terms of how well they actually show like grid cell replay. But we can talk about that some other time.
I should point out that there was so many really interesting references in this paper.
Yeah, there's a lot to read, and of course they're also building on existing models.
Yeah, so
some to dig back into.
Yeah, so this is like a new area for me. So I was going through these papers all Friday through the references, and I ran out of time at the end of the day Friday, so I just ended up printing a bunch more. So I went home over the weekend with about a dozen of these things.
Yeah, I mean, some of those I've already seen because I was talking with Neil Burgess about some of them at Cosine when I was asking him for references for the Nango implementations I want to do in the summer. I mean, who knows whether that summer school in Waterloo is going to happen. But so I think I have a bit of a mental map of what's happening here. But I was thinking we should show briefly the videos because it's going to bring some of these home points a little bit faster and what's happening dynamically here.
I have to do that too. Where are my view options?
It's up top, I think. When you go up to the top, you put your mouse up to the top.
Oh, I see. I have to bring it down. Let's see here.
All right.
1 second. I see mute, stop video, background, new share, pause share. I'm not seeing a view option. I can stop my share. Would that help?
Yeah, that might be easier then.
Do you see a green bar that says you're viewing Florian's screen?
Now I stopped sharing and now I see it.
Right. All right, cool. So these are the videos. So the first one is just basic slanted obstacles. So you see this thing, the agent is moving around from the start to some point and then wants to go back. So vector navigation says go right. Now we're hitting that border. What you will see is that the motor command, these motor cells that they postulate are now modulated by that growing inhibition from the border cell, which you can see. So this is the border navigation, in case you can see my mouse here, right? And that biases the motor cell into a different direction. So there's no remapping here. This is just the biasing of the vector-based navigation by the border cells that sort of change the vector a little bit to allow for that. And because, maybe briefly I should say that, because that inhibition grows with proximity to the border, you get this very nice slanting, which looks very natural, right? Where you get a gradual deflection at first, and then when you're very close to the wall, you get a very tight 90-degree deflection away from the border. So that is quite nice.
Before you go on, Florian, I want to point out, for those of you who know me here, I do a lot of introspection on these things, and you can feel yourself doing this as you personally navigate in the world. It struck me very, very close to how we actually behave ourselves, and it was fascinating just to imagine how, that curve as you get approached. As we get closer to an obstacle, if we see it from a distance, we keep getting closer and closer, and then as we get closer, we start veering off a little bit. We do the same thing ourselves, and it struck me as like, oh, this is amazing.
Yeah, I've been thinking a lot even about the more complicated tasks where you might want to compare, like actually untrained rats. We do a lot of trained rats, but if you do untrained rats and you see them like use these different strategies and switch between different kinds of goal navigation, right? To what extent you can quantify that and verify some of the assumptions of the models or tune them, right? But we get to that.
Before we go on again, I'd like to make another point, and this relates to our research. So, as you know, I've been working on this idea, and we've talked about it numerous times about mini columns representing sort of 1D, a set of 1D basis vectors for defining a metric space. And as part of that, I've been developing further ideas about how the motor representation is represented in each mini column, because every mini column has a layer 5 cell, which is one or more motor cells. And so I could literally visually imagine this process occurring between mini columns, because a set of mini columns are going to represent in a cortical column, are going to represent the motor vector for that column. And you can literally imagine these mini columns inhibiting one another, or cells in layer 5 perhaps inhibiting one another on this very process. So I look at this, I'm translating this back to cortex, and that's pretty exciting to be able to now visualize.
I mean, there's some, I think, architectural problems with much of this. I mean, like they really cut down on the detail in effort to make a more functional model, which is great. So we now get nice videos to look at. I think there's some there's some problem with, for example, this need to arrange cells into ring structures that don't have any biological reference showing them and these things. But we can talk about those things.
My point was, I could dismiss the ring part, but my point, Florian, is I think we can add that anatomical detail. That's what's exciting about it.
Right. Yeah. It's integratable with the columnar model.
Yeah. I wasn't talking about that, but I'm already developing this model of how motor commands might be represented in the cortical column. And so now I have like, oh, this gives me an idea further, like how it might actually work in some situations. Anyway, I thought it was quite likely this strategy is actually what's going on.
So that deflection worked because it was a slanted object. So there was still a gradient of progress. When the object is now perpendicular, that doesn't work. So what you saw there, I'm pausing the video here, was a brief, essentially a sharp wave ripple sequence that activated the marketeer with this triangle, that event, right? And so now we are navigating to, what is it, like 10 place cells before, which is roughly the range of a sharp wave ripple event. I mean, they don't, replay back infinitely. Obviously, there's a range to this phenomenon, but that is feasible. And so you get an intermittent goal and intermittent goal, and then you can resume vector navigation again because you're making real progress. So that again looks, rather natural, right?
I think someplace in the paper, if I recall, they talked about that issue of the fact that is how much replay can you do in one sort of ripple event. And I think there was something about it. Either maybe the author can comment, but it was either like they sometimes they have different durations or they're concatenated together. But they did that issue.
I think there's also an issue to the time scale, because there's theta scale replay, which tends to be like forward, and with this new intricate interlinking with these head direction cells that Edward Musser was talking about, right? We talked about that last time. This pinging left and right, and those are quite different from the longer and more powerful sharp wave ripples, which you get either during slow wave sleep. Or doing stop, consummatory behavior, rest, right? Like what you might get at a T-mace intersection or when you have a perpendicular blocking obstacle. So there's actually different kinds of replay. The model only presumes one, namely playing backwards from the goal location with a long range replay, so like a sharp wave ripple, right? To get a new intermittent goal. Theoretically, one can combine that with a theta forward-looking, like a pinging left and right, what are my options now, to build a merge of these. So that would go into the direction of this stuff that I presented from David Turrell, like a hierarchical planning scheme where you have long-term goal, intermittent goals, and then planning strategies towards intermittent goals.
One thing that I don't think was addressed in this paper is that which I think is fascinating, is this replay of grid cells, place cells. There's several things they pointed out, which are fascinating. One, they go both forward and backwards. And so if an animal stops, I believe, and it goes to this, and so it's only when the animal's not moving, which also makes sense, by the way.
Which is also when the theta stops.
Yeah, so they stop moving, and then there's these brief periods where these playbacks occur. But sometimes the playbacks go forward and some goes backwards. Well, so that's been well documented. So that tells me, it's interesting, that tells you that there's this one-shot learning, sequence memory, but it's bidirectional. And so we don't, our sequence memory has always been forward-looking. Like when we talk about learning melodies, it's you don't learn them backwards, but this system does. And so I don't think there was any suggestion of why the reverse sequence would be useful in this navigation task. Perhaps it is. Maybe the author could comment on that. But this just talked about the forward one. The other thing was you just mentioned there, that the playback of the play cells for this to work, there has to invoke the appropriate grid cells at the same time. And the authors make an argument there's enough time to do that, and they also give that happens. But the other thing I didn't realize is they also make the evidence that the playback of the grid cells can occur without the play cells, that the grid cells can remember the sequence. So you've got play cells remembering the sequence, and you've got grid cells both remembering a sequence. That sort of opened up my thinking a bit about, first of all, the role of sequence memory everywhere, and second of all, the different functional abilities that are afforded. Yeah,
the microcircuits behind the grid cell place interaction are actually quite complicated. It's not that, like straightforward bidirectional connectivity, because all the place cell feedback into medial entorhinal cortex layer 2 is indirect through layer 5 into layer 3 into layer 2. So there's like there's a trisynaptic circuit, and it's a powerful one, and it's everywhere. But it is so there's no doubt about the existence of like feedback loops, but it's not really cell-to-cell connections like what some of.
This is the feedback for which one you're talking about?
I'm talking about the microcircuit data from Menno Witter. I didn't talk about that yet. That's also.
To do what function are we talking about?
To talk about the influence of landmarks, of place cells onto grid cells. So like how would you, during place cell replay, right? Anchor the grid and make it follow along.
So again, I'm always translating everything I think is what's going on in cortex. So to me, if we think these grid cell equivalents are in the lower layers and they're connected, this bidirectional stuff we see between layer 6 and layer 4 and layer 5, then it's not a mechanical problem there. The mechanisms would be existing in the cortex. You're saying in the hippocampal complex that is a more complex circuit.
Yeah, it's a powerful but indirect circuit.
Well, I thought.
Yeah, exactly. They don't have the theta forward replay, like this head direction cell pinging, what I referred to as the sweep yesterday. And what David Turrell then supposed to be like these forward kind of replay theta sequences, which are much smaller. They're like more like 3-4 place cells and not like 10. They're also less powerful events, but they are theta rhythmic.
Yeah, so there's a lot going on, but I think the idea that there's this functional role here is pretty important. I don't know if you want to show this next video before showing figure 3, Florian, because it might make more sense if we walk through figure 3 first.
Yeah, that's actually a good idea. Let me just stop sharing here. Let's go through you showing the paper first, because there's like there's 3 more videos that show all the stuff that the model can do.
Yeah. So we talked about this figure. Let me just go on to figure 3. Here they've introduced some more. These are all simulations again. Remember, there's no real rats here. This is a simulation of the model, and they've made up this this funky little environment. These gray things are obstacles, and the red line is showing how the rat is moving. And so they talk about here where there's a nest right where this red line begins in A1 here, and showing a trajectory of the rat, the animal. And then what happens at this point, then the animal traverses some distance along the perimeter until this is called the start point, and then it's said to get back home. So now the animal's out here where this question mark is and says, well, what's the strategy? How do I return to the nest, which is right at the beginning opening of this little cave here? They go through this in 2 scenarios. They talk about where the return point is right at the opening to the cave, and one where it's deeper in the cave. So you can see B in the middle of the screen, it was at the opening of the cave, and C, it's in the middle of the screen. It's showing up on my screen. And then they talk about this under different scenarios. So here they're saying the vector navigation with obstacle deflection starting from these 2 blue points seems to work pretty well. So the animal heads straight for its nest, it hits this border, it kind of runs around, gets to home. It starts from here, it goes straight towards the X, it hits this border, runs around, it hits this border, runs around, it gets back there. But now they point out if you move the goal, the nest inside the cave, it no longer works because the animals get stuck on the outside here. It's just a fancier version of what we saw before. But then they show in this scenario where you have, we combine the 2 strategies, the animal doesn't get stuck here. it goes back to doing a path integration, not path integration, following its previous, using the topology of the map of the environment. I think in this model, this model was they assumed the animal had a complete model. I forget what it was. Was this this was the environment was understood, I think. I can't remember now. Anyway, so they're showing how then they show what happens from all different points around the circle. So so the animal, they're trying to start from anything around here. And again, as long as the X was in the middle here, then the vector navigation worked. Here, the vector navigation only worked from a few spots, and the animal got stuck most spots. And here, the animal always ended up in the right place at the end. So that was that figure. I don't know if there's a video of that you want to show, Florian.
Yeah, there's a video of exactly this. So if you stop sharing screen, then it's just going to switch over for most people.
I did stop sharing.
Cool. All right, then I can just show the video.
Hey, Jeff, just a quick thing. You may want to turn off your Outlook while you're not sharing, because I think all your emails are popping up.
Yeah, I know. I keep dismissing them. How do I do that? I'll just close it. Yeah. Okay. I closed Outlook.
So here's vector navigation with obstacle deflection. So big running around, and here's a start point, and you just get this all these slanted objects. They don't even need remapping or anything, right? So that works. Now there's a second trial starting from here. And there you have, yeah, then you kind of like get stuck there, right? Because it's a local minima in the energy landscape. And you don't have any way to get out of it. So combined vector placer navigation to overcome that. So you form that place map. You see all these connected places, right? So you have a map. And now you go. And now when you get stuck, you can do replay to an intermittent goal, remap, and get to the goal. So maybe I just play that last part one more time, because there's a lot happening. Right, so first you build the place map by letting the road run around. All these nodes are connected now, which means that we already know the connections here, right? These steps that are important to getting to the nest back. And so you start on vector navigation. Now there's a local minima, so you go into replay because you can't solve it, and then you remap and map and map with intermittent goals through these topological steps. So that was that. Now we can continue with the paper. I'm going to stop sharing.
Okay.
I'm still a little confused how these intermediate goals are determined. And what if the animal's path did not intersect with the intermittent goals? Will it, or is that?
Well, the animal's path will always intersect eventually, because it got to where it is. So I mean, it could retrace all of its steps if it had to, I suppose.
Yeah, but that assumes it can. I guess it can only do it within recent memory.
Yeah, if you didn't have a memory going out of the nest, then there is no way to get back into the nest when you need to use that information of how you got out of the nest.
Mm-hmm.
I mean, in the worst possible case, right, where for some reason, the entire vector navigation, any possible integration is a failure, right? Because you make no progress in the gradient towards the goal. You will just entirely retrace your place map. That is not realistic because sharp wave ripples don't go, like infinite steps backwards.
Yeah, no. So I understand.
But theoretically, the model can do that.
Yeah. So I understand all that, but it seems like the efficiency with which you can return back to your goal is really determined by the exact path you took before. Even though if you knew the obstacles, you could probably, you could step back and figure out a much better way.
Right, but that is not all unrealistic. I mean, like when you explore an environment intensely first, you're much better at taking shortcuts, right? And getting out of deadlock situations. So, I mean, in that sense, you would expect performance to be worse when there's not an extensive place map in existence. Yeah.
Yeah, that's true. But you don't have to have your performance. It's not just whether you have an extensive place map. It's literally dependent on the trajectory you took before.
Yes.
that limits
Yeah.
how well you can get back to your.
Well, if imagine you're out in the woods or you're in some town or something, and you haven't explored an entire section of town, you're unlikely to go over there looking to see if that's a good way to get home, and you're going to look for the surer place to get back, even if it's a little bit longer. So the next figure we'll talk about some of these optimizations, but I think it would be it's kind of unrealistic to ask the animal to go take approach in an area of town where it's never been.
Yeah, no, but it's also, it seems that's true, but that's one extreme. The other extreme is you're literally restricted to the exact path you took before, and that defines, how optimally you can get back.
Well, it's not the exact path.
You're leaving a lot of information on the table that you could use.
I don't know what else you could do. It's not clear to me what, I mean, you're not taking the exact path back. You're using the exact path to find a temporary goal.
Yeah, I
so
think
I don't really understand how these temporary goals are, how they're figured out. And they are dependent on the path you took.
Yeah, the temporary goal. Just imagine if you could, and we could look into their model and how they actually did this, but I'm not sure that's biological.
Yeah, I mean, they must, I think I need to look at it because they must have an algorithm because
they do have an algorithm, but to me, it didn't bother me that what exactly that algorithm was. One could just say, what it looks like they did is they say, as you play back, keep testing whether through vector navigation, whether there's any deflection to get there. That looks what they did. And so the moment you get to there's no deflection, then you say, yes, good enough. I'll head in that direction. And okay.
Right.
Yeah, we're going to talk about that figure next.
Sorry, guys, I was just reading on
at this point, all we assume the animal knows about the environment, and I think what Andre was just saying, the only thing we assume the animal knows about the environment is the path it took to get there, to where they are now. So I think it's a good strategy, given the animal knows nothing else about the environment, is to go straight to the destination, and you get stuck. And if you can't get anywhere, then you pick an intermediate thing that's on the way there, on the way back, and that's the first time you can hit that intermediate goal is when you stop doing the playback and then do another vector navigation.
Can I ask a question on seeking those goals? So I can understand. Okay, you replay it back until you have line of sight to this other, shortcut, but your perception of that shortcut, when you were traversing it first time, object A is over to my right, object B is to my left, and stuff like that. When you're looking for that line of sight, what's the interaction between actually being able to visualize that previous goal point is actually what you're now looking at from a different direction?
So I don't think you want to use the word line of sight, and I think Andre would. If I got this wrong, he should speak up. But there's an algorithm to determine this. What I said earlier, I don't think there's an assumption necessarily that there's a line of sight. It's just what it looks to me the way the algorithm would be working is they're waiting till they get to the point where there's no deflection, and that's good enough. And I think I'm not sure, Kevin, if your question is like, remember the underlying this is the ability very quickly to say from any point to any other point, what is the vector that gets me there? And so you're constantly calculating that from the animal's current location where it's stuck to a series of place locations and associated grid cell locations. As that playback is occurring, it's calculating that vector continuously. We like, this is the and then says, okay, that's good enough. I'm going to go for it. There didn't have to be any prior. The vector navigation doesn't require that you recognize this place earlier.
I think this whole algorithm would work without vision. If it, like whiskers would be sufficient for this.
Yeah. I mean, the
and looks like he replied. Matt posted in the chat. He replied something similar to what I said at the very beginning. That when you get to the obstacle, you know what the angle of that obstacle is as you turn. As when your vector is pointing away from the obstacle and presumably intersects with the path you've taken before, then that's how you can figure out what the intermediate points are.
Right. So that is the algorithm essentially.
Yeah.
So maybe we should go on to figure 4. Is that all right?
Yeah. Let's move on.
Okay.
I mean, you should share your screen again, I guess.
Yeah, I'm back.
Here we go. All right.
Here's figure 4, and I think this is what Andre was just talking about a moment ago. This is looking at this algorithm from a different perspective. This is looking at the algorithm from the perspective of 2 new variables. One is how much of the environment has previously been explored. This is called a dense exploration, versus one where the animal has only seen a little bit of the environment, which is a sparse exploration. And then that's one new variable. And the other new variable is they're saying they're introducing novel or new shortcuts to the system that didn't exist when the animal left. So, you know, I don't remember why they decided to do this, but this is what they decided. So the thing I didn't understand about this, and that's something I really want to get into in the literature, is this idea that you can have the place cells can create a dense sort of map of the, and this can be used for navigation. I don't understand how that happens. You know, so it's just on the assumption the animals explored everything and knows where all the places are. It's just, if anyone knows, you can let me know, but it's something I have to dig into to understand. How is that useful for navigation? But that's what this figure is about. And then they show what happens if you use just the topological agent, this meaning just use your knowledge of places. And I'm not sure if this means that the animals done every one of these paths, or is this somehow there's a map built up? I don't understand that. But if you just use the topological method, what happens in a dense exploration, what happens in a sparse exploration, and what happens with novel shortcuts, and then what happens when you use their new combined their vector and what do you call it, metric and topological method. Here you see under the topological agent, if the fully explored environment, then the animal is able to move pretty quickly right towards the goal. In the sparse model here, the animal can only retrace its steps completely. So it has to, if it's here, it has to go all the way around again, all the way back, and then back into the thing. So that's not very efficient. And also, it doesn't take advantage of novel shortcuts because there's no way of discovering them. It's just going to take its path back. Here, when you use the combined agent, and now they're showing it from all the different points around here, it works. It works well in the dense. It's not taking advantage of the dense model anymore. It's just using vector navigation. Here it's saying if it's sparse, it's using it also works very well, and it's also able to discover, because it's using the vector navigation. It'll discover new shortcuts because it's going straight towards a goal. And if there's a little hole in the wall there, it's, no, there's a hole in the wall here. I can go right through, type of thing. And then they compare the performance of these different networks to showing how efficient they are. And of course, the combined agent does very well, and it's able to take advantage of the shortcuts. It's the only one that really is able to take advantage of the shortcuts, but it does well overall compared to the purely topological agent. So I need to learn, I need to learn how this model works here. I didn't understand that. And then if there's no questions of that, I can go on to the next figure, which I'm going to be very brief on.
Yeah. I did send Andre the link to the Zoom so he could join in directly if he wants to. Why not?
Sure, why not? The forefront. We Andre should know that we're not just doing this because of this virus. We've been doing this recently anyway. So maybe
well, I guess maybe the difference was that I, recently, notified pretty much everybody who was at COSA.
Yeah, so it's great.
Right, or at least all the people at the grid cell meeting.
So I won't spend much time on this figure. It was somewhat interesting, but this was more one of these things like they said, how does this work on 2 types of mazes that have been historically used going back a long period of time, many years ago, and just sort of comparing it to these classic, this starburst, sunburst maze, and this other, which is this, I forget what they call it. What was the name of this one? What do they call it here? The detour maze. And so these are sort of classic in the literature mazes that rats have really gone through, and they're basically trying to show that the model works well on them, and I don't think there's really much insight further into it. At least I didn't get much insight into it, other than they're just showing it's compatible with what mats have done in previous well-known maces. So I promised this would be a fairly short paper. I have a lot of notes on it. I'll just go right to the very summary here, and then I can end it here. This is basically in the discussion section. This is the summary. The agent initially performs vector navigation, primarily driven by grid cells and aided by border cells for obstacle deflection. That's what we showed in the first figure. If progress is blocked by obstacles, the agent initiates a hippocampal replay that introduces aspects of topological navigation to the agent's overall behavior, allowing the agent to switch between different sub-goals in order to successfully complete navigate complex environments. So that's the summary of the paper. I do think there's several things in it that are, to me, and I'll explore this more in a future meeting, how it relates to our models. But anyway, that's it for me. Sure.
It's about, is there any information about rats that don't have an entorhinal hippocampal complex and their ability to navigate in cluttered environments? Do they show any impairment?
Well, yeah, there's lots of literature that shows, I mean, first, every mammal has, entorhinal cortex and hippocampus. So we're talking about ones that, yeah, we have talking about animals where they intentionally disable or in other ways to disrupt this pathway. And I think there's lots of examples, and there's several that were cited in this paper, like what happens to mats in this water maze when you disable the communications between the grid cells and the place cells. There is impairment. Basically, animals cannot find their way someplace very well. They're definitely impaired when any part of the system is interfered with, if that was the question.
Yeah, I think he was concerned about localizing the functions in this complex.
There is a bunch of literature related to that. And there are several of papers referenced here, but I don't know. I'm not familiar with them. I'd have to look more closely. But there are some of the papers that are referenced here talk about that. But I was reading them over the weekend.
All right. I was thinking of maybe showing the videos to the last
if
part.
If you're still there, check your Twitter DMs.
Which illustrates some of the dynamics of this. And then I want to relay some model concerns that I have. Let me share my screen.
And while we're doing this, as I said earlier, I want to go back to either maybe Wednesday or next week or whenever. I wanted to have a more in-depth discussion about current thinking about cortical columns and mini columns and related to this stuff. I'm not prepared today, but
Right.
I was excited about this because he was helping me understand the models I'm working on better.
All right. Hope you guys can see. So combined vector place agent, sparse exploration without shortcuts. So you got this agent which traces out. You see the map of the place cells that got formed, right? All these little place cells. You get deflection. You trace back 10 or so. Like, it's interesting to kind of stop at this point to see that, of course, like the intermittent goal that gets set here, right, gets set essentially on the basis of the gradient, right, when the agent got stuck, right, that border, right, at what angle can I make progress? Technically, one could have chosen one, closer out of the nest, right, but practically it needs to choose this one, and only once it's there can it, like, resume that navigation based on replay and based on topological steps. And you see this nice intermix here, right, where you get the gradient based, the vector based navigation, and then the topological. Now it switches back because it can, right? It's straight line of sight to the target, right? To the vector navigation, a bit of deflection until it gets stuck again and needs to switch again to topological navigation. So there's like this nice back and forth. And then here's one with novel shortcuts. Let's see, how did that one go? You go out. Oh yeah, right. So you see how initially the deflection goes rightward. And that works for reducing like the distance to the goal for a little bit, but not for too long. And so you need to remap and use the existing path out to get back in.
So I'm a little confused. How much has been models and how much has been experimental data in this paper?
This is
So-
All models. Okay.
It's all model. There's no experimental data, although of course they reference a lot of experimental data.
Yeah, it's an entirely
It's like us. We don't do any. We don't do
computational model.
Right. So they're taking all this known things about this stuff and creating models.
This is very much like our columns paper,
Right.
something like that, where we have a model, we model it, and we're trying to make it as realistic as possible, but it's it's trying to be biologically accurate, but it's all model.
All right. And so lastly, the sunburst maze here. It's obviously quite complicated. You get this long path into the maze, and that map you build is useless, at least pretty useless, because now your entrance point is actually blocked. So you can't go back. You actually need to take the goal arm 6. And it's very hard to find it at first, because of course the gradients are kind of pointing in that direction in arm 5 and 4.
Yeah, because it's not super accurate. The animal's going generally the right direction, but you can imagine how if you were in that maze, how you would have trouble too.
Yeah. But so what you see here a lot, right? What is noted in these diamonds is that and these purple things, like when the agent essentially gives up, right, and enters random exploration, so just to build a bigger place map, which may or may not be useful later down the road, in order to hopefully eventually get better at this. And there we find the right arm, right?
So is there some sort of penalty against taking paths that you've already taken before?
I don't remember them describing that.
No, not that I know of. That would be a great question for the author.
Mm-hmm.
I didn't see anything in the model. That would obviously help this model a lot, right?
Yeah.
Yeah, if you could do that, right? Because that is immediately where we go like, okay, this is not
But
real agent.
if you were in that same maze and there weren't any marks, you probably wouldn't be able to remember which arm you went down. It's like, imagine.
Oh yeah. Sure. But I mean, I might even try the same one twice, but not 10 times, right?
Yeah, that's exactly right. And it's still, you still want to do complete random exploration, because that could take still a long. You can do better than complete random.
Yeah. Yeah, I guess there's a lot one could build on top of this model, right, to make it cognitively better. But the interesting thing is then also what is the biological justification for it, and what is the circuit mechanism, and how would that get learned? And so you need to answer a lot of questions on plausibility then if you do that.
Sure, of course, yeah.
So I think in the discussion, sorry, Flor, the discussion, the author talks about combining these with a reward-based model like reinforcement learning, and that would help with the exploration process, right? After the first time you visit, if there's no reward, then you're not going to visit it.
So Andre says no penalty, but a tweak to the obstacle deflection because of the narrow corridors. It's mentioned in the paper.
All right.
Say it again. Matt, say it again. I didn't follow that.
There is no penalty, but there's a tweak in the obstacle deflection because of the narrow corridors.
Interesting. One thing I should point out in this model is, and the authors made it clear up front, is they do path integration with their sort of grid cell equivalent, but it's I think they said it's like a perfect integration. That is, they don't. There's
no noise.
There's no noise in it, and they don't update the grid cells from the place cells. So it's a perfect grid cell system, or a vector navigation system. So there's now ways it's unrealistic, but I think that was an okay purposes again. I'm less interested in how rats actually navigate these things. I'm more interested in these basic principles that came out of it, and I really did like the motor thing. I'll talk about that again in a future meeting. Why I thought that was really interesting, the deflection of the motor vector, and also I don't know if anyone does anyone have a knowledge about how in that fully explored versus sparse model, how the model of place cells how the animal can navigate just using place cells. That was like I didn't.
Yeah. I mean, like the way that they're building these place cells, right? I mean, it's like one-shot learning, and they essentially add a place cell as soon as you're far enough away from the place cells that you have.
How they connected together, navigate.
Yeah, the way that helps in navigation is because they're building like this graph structure. And so potentially, since they do optimal planning in the topological graph, you will get to much better shortcuts when you have explored an area densely, because you can use the place cell graph once you're out of vector navigation.
But the place cell graph is not a metric graph, right? How do I know? I don't even know. It's just because I have a link between two things, I don't know if I'm going in the right direction or the wrong direction. I mean, unless you put it on top of a metric space, that connected graph, I have no idea what it means. I don't.
Yeah, that's true. There's no distances in that, right? That's just relationships.
How do you navigate? How would I know to get? Do I just look for the shortest path between two objects, two place cells? Is that it?
Yeah, I'm sure that's almost definitely what they're doing.
That means you have to do a full exploration. Then you have to search this entire graph structure to find out the shortest path.
Yeah, but they have, right. So they represent the space as a graph structure. Let's see. They say this somewhere here in the paper.
But doesn't that seem biologically impossible, like, to do that?
Yeah, of course. I mean, like, the model has a lot of shortcuts to make it work well without an underlying biological justification.
So I don't care about shortcuts. I want to know if it's even possible, right? So this is a new idea to me, that you could build this. Oh, sure, I can understand you can build a connected graph, but that's basically essentially linking every place or imagine every sensory input to a cortical column to every sequential subject input to a cortical.
Right. I have this the paragraph for it here. So it says the resulting place cell graph reflects the topology of the explored environment and contains sufficient information to calculate the shortest path between arbitrary pairs of start and goal place cells across those explored parts. The graph can, for example, determine which of the current immediately adjacent place fields lies on the shortest path to the destination. So they have an actual implementation of that graph search, essentially.
So either you put it on top of some sort of metric space, or you have to follow all the possible graph paths to their destination and then pick the shortest one, which seems computationally impossible in Neuron.
Yeah, I mean, they say here, right? We implemented a graph search algorithm directly using the place graph structure.
Okay, so maybe I missed that. I was under the assumption this is something that people understood was happening, and I couldn't see how it actually worked. So this to me sounds like it's a bit. I actually don't think it's necessary. As long as you don't try to do the dense versus sparse one, you're basically using the initial algorithm, which is like, I'm stuck, retrace my path until I find a place I can join it and get back in, then I don't need that. So I'm going to go for the moment with the assumption that this is computationally very, very difficult to do this complete graph and to calculate the shortest path. And so I couldn't imagine neurons doing that. So I'm going to.
Well, I mean, they quote a paper here. I haven't checked it out. Mueller et al. 1996, where they say they assume that the hippocampal place cell system can support a similar graph search mechanism, for example, via the resistive network of recurrent synapses, which is apparently some implementation.
Andre suggests that we read that.
Mueller?
Right.
Mueller, was it?
Yeah, Mueller 1996.
The hippocampal is the cognitive graph. I think I printed that out. Did we read that once? I don't know. All right.
I did not read that one.
Just sometimes you get a little disappointed about these things because you say, oh, this looks going to be great. There's a paper all about this. And then you get in there and go, it's a little dicey, I'm not sure.
Yeah, but, I have to say, but it's like you said earlier, it's very similar to kind of the functional models we do. And it's really nice to see them take all of this experimental data, but then attempt to create something that actually works.
Yeah.
The trouble to create all of these complex environments. And yes, to do it, they had to take a few liberties here and there. But, we did that too in our column.
I know. I totally.
Yeah, for sure.
And I think it's totally, but still, it's hugely.
That's how you build functional models, right? I mean, somehow define yourself a cutoff.
Yeah. I'm not sure that this fully connected place cell graph is actually necessary for their model. The early, it's only when they introduced it later to do the talk about the sparse versus, but that to me was less interesting to me. So So this is an area where I said, yeah, they took that shortcut to make this work, but I'm not sure that's necessary to get to the issue.
I mean, a lot of researchers would also argue that like this big construction of like a reliable connected graph, like this idea of how do you learn the environment, that is all like sleep dependent and depends on a lot of sharp wave ripple sequences we play during sleep.
So I thought the exciting part was you could remember the actual path you took. That's great. We know that rats actually do that. You can calculate how to get there directly, although we don't really know the mechanisms for doing that yet. Vector navigation. And those two things, and also the fact that there seemed to be memory, empirical evidence that there's memory of not only of the place cell sequence, but the grid cell sequence. And so a new idea.
Possibly through the grid, I mean, like the grid cell sequence exists because of the place cell sequence. That is the argument. I don't think anybody's arguing for remembered grid cell sequences.
No, it wasn't there. They actually did do that, and there's a paper that says that. That it works. I printed out that paper. I can't remember. You know me. I can't remember the names.
I would be interested in reading that.
It's in one of those things. Here, give me 1 second. I'll tell you what it is here. Superficial layers of the medial entorhinal cortex replay independently of the hippocampus. This is O'Neill, Baccarat, Stella, Schoenberger, and Scrivari. And Baccarat,
Stella.
It's 2017 Science. January 13th, 2017 Science.
All right. Okay, I'll find it.
Can you read that?
Yes, I can.
I haven't read it.
Okay, right. Mm-hmm.
So I thought that was pretty interesting too. It looks complicated. There's a lot going on in this paper. So,
right.
Too many colors.
Yeah, got it.
There's a lot of. It was great. I thought it was, I want to thank Andre again because I think it's a great paper, really well written, and I really appreciate that.
That's really cool when they build functional models. I have a bunch of questions though. Like for example, in order to do that vector navigation, right? They actually maintain two separate grid cell populations.
Yeah.
Right. So
you need to have two locations.
Yeah, in order to calculate a vector, and I wonder a little bit what what like Marcus thinks about that, for example, who has been thinking a lot about vectors and displacement cells and
easy. A couple different easy possibilities on that. One, I'll go with the two different populations. You could say the superficial layers of entorhinal cortex are doing more of like the attentional type that aren't your actual current location. It's more like an attention one. And you could say the ones in the deep layers of entorhinal cortex are more of the thing being anchored by sensory input, and the more of the actual one. That's one perspective on this. Another could say, this is just more speculative, but having at different times of the theta cycle, having being going back and forth between using the same grid cells for both.
Yeah, I mean, I just wonder what is the evidence that there's actual jumping between two different kinds of populations, or that they simultaneously code, right?
We've talked about this a lot in terms of cortical models,
right?
And there's the anatomical evidence. It looks surprisingly suggestive of two different populations in 6A and 6B. And that was one thing. And so that's the first thing Marcus referred to. And then the second one was the idea, and there is some evidence, I can't remember what it is, about this alternating back and forth. And that was also suggestive. We wrote this in the frameworks paper that this might happen. And it had to do with this really weird thing about these layer 5 cells, which are motor output cells, which essentially are a vector of navigation, also feed forward in the cortex. And so one of the ways we speculated that these things could oscillate back and forth. But there is some evidence for, I don't remember what it was, Marcus, but maybe you remember sometime. But there was some evidence for that there's two different phases that would be switching in and out of these two different things.
Another wild card that may come into it is that I've already brought up the layer 2 and the layer 5 grid cells. Now I'll bring up the layer 3 ones. The one place where it does seem that there are reciprocal connections between grid cells and place cells, both directions. At least I could point to the paper from David Rowland where they found them between layer 3 of entorhinal cortex and oh, I'm going to forget, either CA1 or CA3. But there are reciprocal connections, both directions, grid cells and place cells. So that's like, seems to be another population of grid cells. And what's the role of those? There's, in my mind, there is room for something like this, and there are multiple ways it could happen, but I don't know which one's the right one.
Again, just to review the evidence in cortex, you've got, and we saw this from, what's her name's paper, the review paper that
Thompson, layer 6.
Thompson, yeah. She made this point that there's very unusual morphologies of these layer 6 cells, and the morphologies is bidirectional, very narrow, heavy connectivity between layer 6 and layer 4, layer 6A and layer 4, and layer 6B and upper layer 5, I think it was. And they're very unique. They look like they would have this unique function, and they're bidirectional, and there's 2 sets of them back and forth. And so that's been sort of the underlying assumption of our models of modeling objects, is that that's what's going on.
Maybe I'm misunderstanding something. This is a bit of a detour. Maybe I'm misunderstanding something about the model. But one thing I would really like to see is some sort of an inclusion of some kind of like simulation, especially in the regard, like, for example, when you walk up to a slanted obstacle, right? Like, imagine you're doing this. You don't just like keep going, okay, I know vaguely a sort of vector coordinate for my target, and then I run into a slanted obstacle, go like, oh no, 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 and then like sort of, take a gradient to avoid it. You sort of have a model in your head about the limits and the orientation of obstacles and stuff, and how you would work around them, even in like only partially, sparsely explored environment.
I'm missing that, Iris. I'm missing that. It seems so. I think in this model, the animal does not know about this obstacle, right? There's no prior knowledge about it. It's like you're running through the woods, and all of a sudden there's a tree down across in front of you, and you have to go around it. You tend to go around the way that's sloping towards the place you're going. I don't think there's an assumption or a model about this, if that's what you're asking.
No, exactly. So that's what I would like to add that as an assumption to the model. I mean, okay, maybe this works better for, like, if you have a line of sight that is significant, and I guess if you're the size of a human being, then that would apply. Or if you're like a rat and you can't see the like a log in front of you until you're right next to it, then that might be different. But I would like to include something like sort of more like a larger line of sight kind of planning.
Well, I mean, there is no line of sight here, right?
Why would it be different? I mean, basically they're saying that at some point you detect this barrier. It's before it's before you don't run into it and it cuts you in half. I mean, you detect whether it's whiskers or you got your hand out feeling it, or you got eyes, or I don't know what it is, but there's some sensor that lets you detect the barrier. And at that point, that's the first point you can start deflecting. Obviously, if you're using a whisker, then it's going to be the distance of a whisker. Can't be seen.
If you're using the eyes, it's quite a bit further,
yeah, exactly. I was thinking more along that range, and then like thinking of like, oh, okay, I just have like this sequence of places, of place cell activations that I had recently, like that I can replay through, and I'm just going to go back there and plan another route. That seems like it's a cute model, but it seems a little bit naive to me. Of course, like, it's great that they made this work and everything. I agree with.
Why does it seem naive to you? Because I felt it struck home to me like, oh my God, this has got to be right. Why did you think it was naive?
Well, so for example, you said like if you're in a new city and you don't know where to go, you don't try to take a shortcut. You go back, you retrace your steps. I think partially that's because of risk aversion.
That's not. What do you mean? I mean, if I'm in the city if I need to get someplace, I'm in New York and I need to get someplace. I know generally the direction I have to go, and if I get to intersections, I have to make a decision: do I go down the avenue or do I go up the street? But I think it works in the city as well as it does in the forest. I'm not disagreeing. I just don't understand your objection.
Right. I'm just saying. I think maybe the sort of the gist of the paper might underestimate our capacity for like building a model of the environment and our ability to interpolate through it. Maybe I'm being unclear, and maybe I need to read the paper in more depth. But
yeah, this is sort of, it's a little bit similar to kind of my concern as well, that you're limited to what you can sense right now and the exact path you took before. Those are your kind of the 2 bits of information that you're working with, and you're not trying to build any sort of a higher level model or anything else.
There's no flexibility beyond that.
Sort of probably beyond the scope of what they're trying to do. I think that's a whole different level of stuff.
Yeah.
Well, absolutely, yeah. But it would be something nice to sort of start thinking about if you want to work with that.
Yeah, no, I agree. Yeah.
Yeah.
One thing that I've been thinking a little bit about in terms of, biological evidence for these things, obviously, there's a lot of, like, it's very nice that they're using sort of that evidence for grid cell replay and that coordination between place cell replay and grid cell replay to like make it possible to do this, fluid switching, right, between vector and location thing. But the way that they built the place map also, like, I mean, in biological circuits, right, these are contingent on, I mean, Elafit, right, would make arguments about the capacity of the place cell space. You can't just put a place cell anywhere. And people have been looking at replay and pre-play, right, also say that, well, look, I mean, these cells are not really like new and then added to the sequence. Actually, they're kind of in the sequence before. They just get recruited as representing that space. So like, all this evidence from pre-play that sequences of certain cells exist before they're ever being used for mapping a particular stretch of the environment.
These pre-plays exist before the animals actually navigated that direction?
Yeah.
I mean, there's an infinite number of pre-plays.
Yeah, yes. Well, no. It turns out there's only, so if you look at replay activity, right, there's often sequences of cells that have no particular like behavioral relevance in this environment, but they might get recruited later on.
Did the animal navigate them?
Yeah, like doing.
Okay, so forget the relevance part.
Yeah.
My point here is, I actually think this. I mean, the relevance is obviously going to be important, but why I walk away from this is that we remember this stuff completely, even if it's not relevant, and we'll forget it again quickly if it's not relevant. But you don't need relevancy to remember this. You're going to remember the path. I wrote about this in the book about going to get ice cream in the kitchen. And you're going to remember the details of your walking to the kitchen, even though it wasn't relevant, but you'll remember it for a very short period of time. You'll remember exactly where you went, what you saw along the way, but you might forget it in 10 minutes.
Yeah, we have a very detailed memory for stuff that's happened very recently in the past.
Yeah, so the idea here is this constant one-shot memory of every little thing that's going on, and it just doesn't last very long unless it's got relevance. So I think.
Now, I guess my point is, I'm not disputing that at all. I mean, I'm all about working memory and fast associative heavy and plasticity. That's not what I mean. What I mean is that the actual mechanism for selecting that cell is not free. So you cannot loop any place cell into any sequence. It's kind of the other way around. There's already a cell that is likely to be part of a sequence, and then when it's needed, it's recruited into.
So the way I've always looked at this, and the way I've talked about it, is that place cells are inherently sensory driven. They are the sensory, and so they're not picked from a pool. They're not just randomly chosen to be here. It's what would occur here by your sensory input. So at any point in time, and look around, you see something, you know where you are, you're going to invoke some set of place cells based on what your actually sensory experience is at that point in time. So if you think about it that way, then there's always a population that'll be active at any point in time, as long as you're sensing something.
Yeah, sure. But people like Elafit would argue, look, that the evidence for the way that place cells get recruited, right, suggests that the amount of actually like realizable places is a lot lower than the amount of cells, right, like would suggest.
But of course, but that's not true, because if you look at a population, like you're doing a population code, you get 10,000 cells and you pick out some 200 at a time. There's an infinite number of combinations. And place cells do repeat, just like grid cells.
But theoretically, but not really. So when you induce place cells, for example, like the way that Aaron Milstein did, right, with the dendritic plateau potentials, then you don't get a narrow place cell at the exact moment that you put that pulse down, but you might get a place cell 3 seconds before that or 2 seconds after, somewhere in the neighborhood here. there's going to be one cell that responds and it's going to get recruited.
So I guess this idea that there's one cell doing anything is ridiculous as far as my concern. It's always a population. And if they're measuring one cell, they're only measuring one cell, but there's always going to be a population. Otherwise, you run into grandmother cells as place cells, and that's not going to happen.
Right. Which goes to my point of contention, that they are just like adding a new place field whenever they need one, as opposed to a more realistic mechanism of how they get samples from an existing population.
All right. So I don't know what they did in this model, but if it's single cells, yeah, I agree. That's not right. So when I looked at the places in the place cell chain, to me, it's always, that was like our sequence memory. That was a series, a sparse activation out of a larger population of cells. There's a virtually unlimited set of those, and you can learn quite long sequences of those. Thousands and thousands of elements very rapidly.
Yeah, I know, it's very impressive, the capacity of that. Yeah, but it's on a pool of existing cells, right?
Yeah. So I think anytime, maybe I'm wrong about this, but anytime anyone talks about place cells, I'm always thinking it's a pool and you're doing a SDR of place cells. There's never the idea that there'd be a single place cell representing something, to me is crazy. That would never be the case.
Yeah, I think it's actually like an artifact of the way that we record. That we used to have tetrodes. I mean, maybe it's going to change now. Maybe, sometime in the future, now that we do these dense recordings with the neuropixel probes, you stick a bunch in there and you're recording 600 cells. Maybe that's going to change, where we're no longer talking of a place cell, but we're talking about, a place assembly, right, or something.
Yeah. So imagine this. Imagine you have, you have a 2% activation in your place cell code. And so now you have, to find one of those cells active, it's 1 out of 50 chance, right? But to find two of them active at the same time, it's 1 out of 2,500. So it gets really hard to find two active at the same time. And it could even be 1%. Then you go up to 10,000. So it'd be very easy to miss these coincident activities, but it's almost a guarantee that it's happening. There's nothing ever done in the brain in single cells. Yeah. So I just assume that's the case, and I assume that people don't see them because it's hard to see.
I mean, it's also just one of the simplifications that the model makes in order to stay minimal and to, not confound the stuff with too much detail. I just wanted to point that out, that, if I wanted to build an implementation of that, I think it would look different.
So in our models, Supertaylor was working on this paper that we actually ever published, which was showing how the same neural mechanism that we, for our sequence memory, works for sensory motor learning as well. And so in one case, the sequence memory, we take these inputs and we just learn the sequence as we go through it. In the other case, we're trying to pair it with a motor signal, a location signal from what would be grid cells. And what I walked away from this is that I think the upper layers of the cortex are learning every single transition, no matter what it is. It's a sensory motor sequence, which is a very unique thing. I may never do it again, but it could be forgotten in a little bit. Or it's a high order temporal sequence like a melody, and I'll remember that too. So now we've really unified those two ideas. You can say, okay, sensory motor inference and learning, and it doesn't matter how you're doing it. It's going to go quick one-shot learning, all that stuff.
By the way, that the paper on that thing is on bioRxiv.
Yeah. I guess it's published. we gave up on getting whatever journal.
That's as published as it's going to get.
Yeah. All right, I think we've gone on quite long here, unless someone else wants.
Yeah, I need to take a break before our next meeting. So our next meeting.
Right.
Okay, guys.
Yeah, it was a good chat. Super interesting paper.
It's a really cool paper, and thanks, Andre, for listening in.
Yeah, thanks, Andre.
All right, I'm going to end this meeting, you guys. I'll see you at lunch. Okay. Hey, it's just you and me now. So I wanted to say something real quick. If any of you guys are watching and you're like, why do you guys care about grid cells, place cells, environments, and stuff like that? So the Numenta's theory of intelligence involves the mapping of space as we learn objects with our sensors, and that's what this is all about. Grid cells, place cells, mapping space with your sensors. And we think these same mechanisms are used not just in entorhinal cortex, hippocampus, but we believe they're also used in cortex, and that there's a circuit in the cortical column. There's a bunch of different layers in there, and we think that there's grid cells and place cells operating in some of these layers in specific ways and are wired up to other layers in a sensory motor sort of loop. I don't know if loop's the right word, but we're trying to understand all of how a model, we're trying to create a model of how this cortical column works that includes the ideas that we've understood grid cells and place cells to work upon and how they map space, because we believe that concepts and objects are essentially the same thing for your brain. So if we can understand how an organism can map a space with its sensors, whether it's running around the space or whether you're holding something in your hand and touching it with your fingers, then that will help us understand how we can map conceptual space and understand how thought works, how concepts can be organized and linked to other concepts and other things. So this is a core sort of focus of our work at Mementa. So I just wanted to go over that so you understand why we care so much about place cells and grid cells, because these seem to be mechanisms that are mapping space. And I think that they are really important. We all think at Mementa these are really important to understand, because we think they're happening not just in entorhinal cortex and hippocampus, but all throughout your cortex, all throughout your neocortex. And they're involved in the representation of everything that you have an understanding of. And that's it. Thanks for watching. We will be back Wednesday. We'll do another live stream. And I think Florian will continue talking about cosine in his third installment of his cosine recap, which is tons of information. It's a good series, so check it out. Be sure to like the video if you're watching, and subscribe to our channel. Take care, everybody. Have a wonderful day. I'm going to hit the stop button and head home and check on my kids who are not in school because of COVID-19 scares in California. Stay healthy, everyone.